So welcome everyone. Thank you for joining us today for our podcast on how to avoid common pitfalls when advancing from formulation development to preclinical testing. My name is Julie Forgette, and I'm Senior Director, Safety Assessment at Altasciences. I will co-host the discussion with my colleague Andrew Buis, who is the Senior Formulation Scientist at our CDMO site, where drug formulation, development, and manufacturing takes place.
Hi, Julie, and hi, everyone. For those who are not familiar with Altasciences, we are a fully integrated CRO CDMO with several sites across Canada and the US, offering biopharmaceutical companies early drug development solutions, including preclinical safety testing, clinical pharmacology studies, bioanalysis, research services, and manufacturing and analytical services.
So Andrew, let's talk about formulation to start. What are clients' typical formulation needs at the very start and then while transitioning into preclinical trials? And how does our CDMO team support them?
Sure thing. When clients first reach out to contract our services, they have an end goal in mind, but don't necessarily know how to get there. For example, a client will present us with an API, often highly potent, its effectiveness as a drug, a target dose, and the desired route of administration. It is the duty of our formulations team to develop a formula that meets all the client's parameters and objectives. We have formulated, tested, and manufactured almost every pharmaceutical dosage form on the market, including liquid and powder fill capsules, over-encapsulated capsules, nanomilled suspension, solutions, creams, gels, powders, and terminally sterilized injectables. We manufacture these products in our grade C and our grade D clean rooms.
I can surely relate to that. In the preclinical field, we work with the manufactured raw material, such as the powder and liquid forms, but rarely with the final clinical dosage form that will eventually be administered to patients. We generate suspensions and solutions by diluting the test material in a suitable vehicle to meet the intended concentrations that will be used on a safety program. For non-clinical safety testing, we prefer liquid formulations. The main reason for that is because at this stage of the drug development, we often encounter technical challenges associated with the selected species when it comes to the route of administration. So for example, oral gavage of a dose in rodents is a standard administration method for an oral drug because we would not be able to dose a capsule or a pill due to the small size in relation to the anatomy of the mouth. Most of the time, sponsors come to us with a vehicle already selected. Our team of scientists then works closely with them to confirm if it's feasible based on the tolerability of the vehicle for the selected species and route of administration. Classic examples of tolerated vehicles are MC for oral suspensions or saline for injectables. We had a recent example where the test material was provided in the form of a gel for an oral administration in rodents. So we did a few tests to see if it could pass through a needle gavage, and then it became evident that it was too thick. So the vehicle was modified slightly to increase its fluidity, and the program was completed successfully. So Andrew, take us through what happens next once the client API has been formulated.
Sure. Once the client's API has been formulated with all the criteria we are looking for and all the necessary testing has been completed, we are ready to begin the next stage. Following all the paperwork and filings, we'll be ready for preclinical scale-up and manufacturing. Because we are an integrated CRO and CDMO, our teams work very closely together, and the transfer of the projects from one team to the next is seamless. It helps a lot that our sites are also located in close proximity with each other.
That's very true. But at times, challenges come up during this important transition phase, going from benchtop to preclinical testing. Can you name a couple of issues that you've encountered and how they can be mitigated?
So one of our biggest issues when going from research and development to GMP manufacturing for preclinical testing is scaling the formulation and associated processes. There is a big difference in designing experiments on the benchtop compared to preclinical manufacturing. As such, one of the best ways to solve this issue is to picture what the process looks like, scaled up, and apply as much as you can to the lab scale. This helps mitigate any glaring issues, but scaling up testing is also required for fine-tuning the process. However, this can be particularly troublesome when large batch sizes, as you will need larger capacity equipment. Another issue that we tend to run into when going from the lab scale to preclinical manufacturing is sourcing material that can be used in GMP manufacturing. These are materials that are produced under controlled environments that follow good manufacturing practices defined by the FDA. This issue can be avoided during the formulation development stage when excipients are being screened. This can be done by researching and selecting more common excipients used in the planned dosage form, as the more common excipients have multiple GMP grade sourcing.
Well, thanks for the tips. I'm sure our clients appreciate it.
My pleasure. The more prepared you are from the start, the less roadblocks you will encounter during each phase of development. So, Julie, let me turn the question back to you since you're the preclinical expert at Altasciences. Once the preclinical team begins safety testing with the client's formulation, what happens then? And what are some of the common pitfalls you encounter in this stage?
Well, in my experience, the main challenges we encountered in the preclinical testing phase are scale up between benchtop formulation and preclinical batches, and communication of special instructions or specific physicochemical properties of the test material when it comes to formulation preparation. Scaling up may result in the suspension not being homogeneous anymore. Another example could be that we see visible particles. We see particles in solution when a clear solution is expected. This may be due to a different reason. It could be precipitation of the test material or precipitation of the excipients. So those unforeseen events may generate significant delays in the initiation of the preclinical phase, as additional formulation development will be needed. In terms of communication, well, communication is key to a successful study execution. The sponsor and the CDMO are the most knowledgeable when it comes to formulating the test material for preclinical studies. They should provide the preclinical CRO with as many details as possible on how to formulate the material into a dosage form. We want to foster a partnership with the sponsor and learn from their experience on how the material behaves. Is it pH dependent? Is it light sensitive? Is our lab equipment suitable for the preparation? Is the material compatible with the dosing apparatus, syringe, needle, and infusion sets? Those are some of the critical details that the formulation team needs to be aware of to ensure a successful preparation. So I turn the question back to you. How does your CDMO team overcome these challenges?
Many of your questions can be answered during the formulation development phase. Even before any lab work can start, we must research the excipients and packaging components needed for the desired dosage and delivery that are easily scalable or have desirable stability profiles in larger quantities. It is also a good idea to have alternative material if information is limited. To help with this, there is an FDA database for inactive ingredients that are generally recognized as safe. It lists many common excipients that have already been approved in other drug products or patents. Once the excipients are selected, we run compatibility studies against the API. The data generated by these studies, along with the API provided by the vendor, allow us to predict to an extent how the API will behave once in dosage form. So how do we handle unforeseen issues that arise during scale-up? Typically, it is not a good idea to rush into manufacturing because any issues that arise can lead to setbacks. When scaling up, pilot batches are necessary to help identify these problems and will lead to a smooth transition into preclinical manufacturing. This allows you to establish a baseline for manufacturing with increased surface area and volume from the larger batch size, from which you can fine-tune the manufacturing parameters such as heating and cooling times, mixing and homogenization speeds, hold times, and order of addition of the materials. Optimizing these factors gives us control over key factors like viscosity and solubility. If issues continue to arise, then we may have to look at tweaking the formulation to address the issue.
Well, Andrew, thank you so much for sharing your knowledge and expertise with our listeners. I'm sure that all these tips that come from years of experience will definitely help our audience during their early drug development. Can you let them know how they can reach out to you for any additional questions regarding drug formulation, development, and manufacturing?
Thanks, Julie. All I can say is whether your API requires a simple or complex formulation, preparation is key to saving time and money. For any questions, you can reach out to me directly using the email address on the screen.
Thank you all for listening. Be sure to check out our other podcasts, webinars, and videos on Altasciences webinars and podcast channel. Talk soon.